Just make sure that some of you were not here the previous day, so your activity is recorded in the all classes, so just remember that. And I have a phone on the podium, so you're thinking that what I'm doing, this is my timer, just to say that I'm not browsing or doing anything. I mean, like a lot of people, it's like you're telling everybody to put away their phone, what I'm doing with the phone. Anyway, so couple of announcements, and I have to make some adjustment. I can't, so this first slide is all about that. So the first thing is that a couple of you were missing the first class, you were not here, and read the syllabus in between lines, because I'm just already getting some of the questions which are clearly written in the syllabus. And again, if you are in doubt, I'm not saying that you don't email, but there are some redundant questions that you will find in your syllabus first. So I'm changing the course participation policy. So it will still be there, but the problem is that I'm just getting these hundreds of emails and my TA, and he is like, I cannot do my research. I'm making up Excel files all day long, which day who will be absent. So I have to modify it a little bit. I don't know how Deborah Ragland actually does that. She has the same way, same type of policy. So here, the class participation points will be dropped for 5 days. That is 20% of the absence will be allowed, and you do not need any excuses. That is think about this, 4 classes means 2 weeks, 2.5 weeks. If you are missing more than 2.5 weeks, again, if you have any other type of issues, that's a different thing. But just make sure that you have that. And the only reason that you need to talk to me is the rescheduling of your exams due to conflicts, or when you're traveling somewhere. That is completely excused. That doesn't fall into this category as such. So if you're missing on an exam day and you're taking the makeup, those days will not be considered. So other than exam days, the other days, I will just drop 5 of them. It still be counted 5% towards your grade, but this will just actually reduce, and I will have a little bit of more time, and my TA will have a little bit of more time to doing their research rather than just reading your emails. And there are some fake ones. It's kind of funny, but that is okay. But I'm not focusing on those, and there's really no reason for wasting time on that. Now, this is another thing that the honors and the graduate students, just excuse me, the others, please do the wiki training and the registration. We will have the scientists choosing the next week. We are not doing it today. There's a reason for that, because if you can see, there are only 2 people out of all the honors and the graduate students, close to 25, have registered. So none of you even have an account. So there is no reason. First, you have to go to the wiki to understand what you're doing. You take the training. After that, building the page for the scientist who is a cell biologist comes in. So do this by this weekend, and then we will go with that. So I just blacked out the names. There are only 2 others who actually got into that. So being said that, that's pretty much the basic announcement. So again, remember that up to 5 absences, you do not have to ask me. Don't send me any emails or anything like that. That is absolutely fine. You do not even have to ask permission. If you're just not absent on one day, that's good enough. So that's fair. Any other questions as such? Any informal questions? And one thing, I apologize if you saw my note in there, that the first week I always fall behind, because there are every day, all day long, there are meetings, and you will have your PowerPoints and the videos uploaded before your class, not like at 3 o'clock in the morning. The last video I uploaded like 2:45 in the morning. So it's not going to be that bad. So it's going to improve. So let's start with the material, and we have already talked about the regular light microscopy. We're going to cover the fluorescence microscopy, and we'll do a little bit different way, that in the sense that in the last class we actually talked more about most of the slides, and we're going to do a little bit different way, because I don't think that there is a reason for repeating all the slides as such. So here, just giving you a couple of pointers in here, that you can see that the fluorescence microscopy, what's the advantage over the regular microscopy, is that you actually have an excitation wavelength that you actually excite the fluorophores. And what you do in this process, that they will absorb a specific wavelength and they will emit a different wavelength. And that's the principle of the fluorescence microscopy. And you use a series of filter to specifically see a single color that you actually want. So that's basically what's being shown here. So you can see right here that this is the green fluorescence due to the excitation lambda is 492, which is in the blue range, and it's emitting a green color right here. And it could be red, yellow, many different colors. And you actually block all other colors and only this color shows up. Now, why all these colors comes in? So remember that in a cell, when you are trying to do this, these excitation wavelength 492 nanometer or whatever it is, it could be many different wavelengths, it's going to fluoresce other things, which is not stained with any fluorophore. Many of the proteins are actually autofluorescent. That means that they will absorb different wavelengths and fluoresce something like that. And you have a blurred image and you actually block all of these so that you only see the color that you want. So that's the principle. And how you do this, just an overview of that you actually do have an excitation filter from a light source. And you actually get a monochromatic light. Monochromatic means it is just one specific wavelength, 492, 492, 512, 512. Very specific monochromatic light, good optics. And then there is, in the heart of the microscope, there is a dichroic mirror. See that this dichroic mirror is very unique. It actually reflects certain wavelengths and allows to pass through certain wavelengths. These are some kind of, like you have seen, those mirror type of shield where it looks like a mirror, but like a helmet shield or something like that. It looks like a mirror, but you can see through from inside that. The dichroic mirror is something like that. So what it will do, that from this excitation wavelength, it's going to fall onto the specimen, and it's going to emit the fluorescence wavelength, which will pass through the red light, pass through the dichroic mirror, and you can record that. And you have a filter right here. You have to have a filter, which is not shown, but the filter will actually specifically, which is the dichroic mirror, specifically cut out all the wavelengths except the excitation wavelength. So that's it's all about. So the next, what you're seeing, that you are looking at a cellular component. Here, just one thing that when you actually, throughout the book, you will see so many color fluorescence images where you will see these different colors. Always remember that you cannot see the green and this is a take-home message. You cannot see the red and the green at the same time. Not with your eyes, not with your computer. So, not with your camera. So what happens, you first actually set the filter, which is the emission filter, to the green, and your computer recorded the image for the green. Then you change the filter to allow the red, and the computer actually allows the red to be recorded, or your eyes. Now you merge those two images to get this multicolor image. You cannot, because think about it, if you have a filter which is allowing green, it will not allow, so just this principle right here. It will allow the green, but it will not allow the red. When it allows the red, it will not allow the green. Otherwise, the purpose is gone. So each time you can only see one. You see that multicolored, beautiful images, but they are taken separately and merged together as such. So just remember that. Here is a variation of these microscopy. You can see that this is a migrating cell, and the dye, which is a Fura2, is actually a very unique dye. What you do with that, again, there are lots of details in your book. You will see that. Again, you really do not have to go to too much detail in that. I mean, like, I'm just going a little bit off here. Someone actually asked that you say so many different things that do I need to write down everything? Just remember, I'm just reminding you that if it is not in your PowerPoint as well as the book, which I discussed in the class, I say a lot of different things just so that you just understand the material. It will not be in there. Say, for example, I say the color of the media, or say ears are being produced. I'm not going to ask any of those questions in there. They are not on the PowerPoint or something like that. So I will be fair on that. So I mean, like, really, for your understanding, you may have to take some of the notes. But again, if it's just the overall understanding, you really do not have to take extensive note. Pretty much everything is either in your book chapter or in the PowerPoint. So this particular Fura2 that dye, it has actually a couple of carboxylate groups which you actually bind with the ethanol and make an ester, and it passes through the plasma membrane into the cell, this dye, Fura2. And then in the cell, you have esterases which actually breaks down this ester, and it actually binds with the calcium, and it's calcium dependent binding. So it will fluoresce depending upon how much calcium is binding. So here it is actually moving in this direction, and you can see that there is a low calcium concentration. In this, probably there is some chemoattractant, and this is actually started moving on this direction. And you can see that when they actually do move, calcium has a lot to do with the cytoskeletal elements which remodel constantly. This is your actin, myosin, and the microtubules. They remodel, and these changing of these microtubules, or the actins and the myosin intermediate and the microfilaments, actually need a lot of calcium activity. So here you can see that the calcium is increasing. So this is the overview of that. Just the essence of the experiment is needed. That's it. So here, this is a very important concept, so definitely remember that, because we will use that. So we'll spend some time here. So now in the variation 2, this is your immunofluorescence microscopy. And what we are trying to do, I just skipped one slide, I guess. Yeah, sorry about that. So here, we are trying to actually raise an antibody against the protein X, because what we want, this protein X is present in a cell. I want to study that cell by immunofluorescence, so I need to raise some antibodies against that. I inject a rabbit. You can inject a goat, mice, llama, anything like that. And this is, they are going to raise the antibody, and remember that these antibodies are called polyclonal. They are not monoclonal that we talked about. That means that you inject the protein X. Protein X has many epitopes. It's going to raise all the different antibodies to all the epitopes, and it's a polyclonal antibody, and that's good. And you may have to purify them after that. It takes about 8 to 10 weeks to develop the antibody. You need to probably add the protein 2 times, means like you have to give a booster injection after probably 4 weeks and 6 weeks, and then you check the antibody in the serum of the animal and isolate from there, and you have to purify some routine things, and then you actually get the antibodies. And if you're not gone into the immunology, I'm not going into the detail of that. This is a typical antibody structure. What you are seeing in the antibody structure, that it is a Y-shaped molecule which is called an immunoglobulin, and there are many different types. There in immunoglobulin is called IG, and it could be IgA, IgD, IgG. So IgG is the most common one. You do not need this information. So all these IgA, D, E, G, these are called the isotypes. And the most common one is the IgG. And a couple of them have a different variety, but most of them have a basic structure, which is a Y-shaped structure, made up of a light chain and a heavy chain. And what you are seeing, that FAB, that fragment, is actually essentially the red areas are the antigen binding sites. So the epitopes actually are bound to these areas. So it binds right here. On the other hand, this black area, which is the FC, is constant. That means in the polyclonal antibody that is going to be raised in the rabbit, there will be a lot of this FAB, or the antigen binding region, will change because it's polyclonal antibody, many different types. But all the IgG, that's the subtype, the FC region is exactly identical in all the antibodies that you get from the rabbit. So that's the essence, that FC region is constant, its shape doesn't change, but the FAB will change. But right here, after you can purify that, this is your protein X, and the antibody will bind to this with these Y-shaped structure. Remember that this doesn't bind the protein. These are the binding regions which are red in color, which is antigen binding. So this is how you first raise the antibody. So after raising the antibody, we start from here. There's a lot of information in this slide. So here, suppose in the cell, this is the protein X that you want to study. So you fix and then permeabilize the cell, because the antibody, large molecule, it will not pass through the plasma membrane. You need to use some kind of acetone or ether to make the lipid go away, and then it can enter the cell. Then it will bind to your protein that you raised against. Now what you do, that this secondary antibody, this is called the primary antibody. So your primary antibody binds to the protein of interest that you raise the antibody against. And you raise the primary antibody in rabbit. Now, what you do that you have a secondary antibody, and this secondary antibody is raised, again, very carefully understand if you are not familiar with that, the secondary antibody is raised against a different animal against the primary antibody of rabbit, not this one. So the IgG, or the primary antibody that you have used, you take just a repertoire of IgG from one animal, that is rabbit. If I just put that in a goat, I actually have a goat anti-rabbit antibody. Because remember that you cannot have the secondary antibody from the same animal, because if you inject this antibody into the rabbit, it is its own protein. It's not going to raise any antibody against it. So this is the concept. Your rabbit antibody injected in rabbit antibody will do nothing. It's its own protein. So you need to inject it to a different animal where it has a little difference, like a goat or a mouse or a llama, and it will raise the antibody against this specific antibody. So you have two of them. So the secondary antibody, which is raised in a separate animal, is conjugated with a fluorescent dye. And then what you do that you apply the secondary antibody, and what you are seeing that this is commercially available, because no matter what, all your single animal, which could be rabbit or rat, the FC portion of all rabbit antibodies are the same. So if you just buy, say for an example, this is a fluorescent, like a green fluorescent level. So if you just buy commercially anti-rabbit antibody conjugated to a dye, that's all you need. And all the labs can use the same thing. Whoever is using a rabbit antibody as a primary antibody can have an anti-rabbit antibody. And this could be a goat anti-rabbit antibody. That means it's raised in goat. You can have a mouse anti-rabbit antibody raised in mouse. So this is a very important concept. Remember that. And you can see that it can, the multiple antibodies can, secondary antibodies can bind to the FC region, and now you visualize them under the fluorescence microscopy. So why you use the secondary antibody? What happens that making these fluorescent conjugated antibody, you will actually destroy almost half of your primary antibodies. You can tag your primary antibodies as such, but it is a very time-consuming process, and you take 8-10 weeks to raise this antibody, and it will take the purpose away. Whereas these are commercially available, and that's the reason that's much more cheaper and much more easier. And the second thing is that as the primary antibody is bound to several of the secondary antibody, so it amplifies the signal ratio. So that's an important concept. So this actually just shows that how you actually put the first primary antibody, and you can see the secondary antibody is bound to the primary antibodies, that FC region, which is a constant region, and this you can visualize under the microscope. So these fluorophores could be red, green, blue, whatever you use, and it's a routine chemical process to add these to these antibodies. So here are just a couple of examples, and I'm not going into the detail of each and every picture. These are very well explained in your book. These are your book pictures. I have uploaded those videos. So here is a glucose transporter, which is not present on the brush water. So this is actually an apical surface, which is actually towards the lumen of the intestine. On the other hand, this is a connective tissue. This is towards the basement membrane. So you can see that this transporter is present on the lateral and the basal surface of the cell. They're tall cells. They are on the side and the base, but they are not on the top, which is facing the lumen. So this is actually a distribution. You can see this in this process. So your question could be, so your primary secondary antibody or based observation is this yellow thing, that what you're seeing. So your question could be that why is this reddish background? So this is actually stained by something called Evans blue, which actually gives you that diffuse background. Otherwise, in a complete black background, you will just see this U-shaped structure. This gives you a kind of a diffuse red background to just have a good contrast that where the things are. without compromising the quality of the image. So here is a modification of the technique, which we call as a double immunofluorescence. So here you have a tubulin, and it is bind with a primary antibody attached with a secondary antibody. Same. You can do the same thing with this actin as well. But here, instead of doing that, you can have a modification. See what has been done here. This is a rhodamine, which is this red dye, is labeled with phalloidin. And this is actually a protein which actually, this drug actually binds to actin. So you can have lots of variation. You can use antibodies to detect that, or if a drug directly binds to a particular structure in the cell, you can conjugate a dye and directly actually apply that. So you can use combinations of different. This could have been labeled with antibody as well. You have that option. All right. So here you can see some classic picture of these. So you again remember that you have taken this picture first, or separately. You have taken this picture separately, then you merge them. Right here you can see that there's some nerve cells, red. Then the nuclei is the blue, which is a hoaxed dye. It's a proprietary dye, and the cytoskeleton green color. You can have 4 or 5 different colors. You can stain all together, take those pictures separately, and you can merge them together. As such, if you just go to the imaging facility, they usually every year have a competition. Have you seen any of those pictures? They usually will have a competition somewhere in the later fall. You can go and see stunning images. It's a competition all throughout the world. So they give these images, and it's amazing to see. All right, so here there is a little bit of modification, and this is a good technique because the antibody raising is a lot of work. You have to maintain the animal, you have to feed the animal, you have to feed the animal during Christmas and New Year break as well. So seriously, it's an issue as well. I mean, like sometimes your animal facility will tell you that during those break you are responsible for that. No one is there. So here you have an X gene, say for example, which actually produces this X protein that is expressed in the cell, and you want to tag that. So without raising the antibody, what you can do, you can actually add an epitope to that. And what are these epitopes? There are many funny names called FLAG, F-L-A-G, MYC, M-Y-C, then HA, which is from a hemagglutinin from influenza virus, HIS, 6 histidine residues next to each other. So these are small, short peptide sequences, which are different epitopes. And you can actually, by recombinant DNA technology, attach that short sequence of the peptide, which is an epitope, many different commercially available, to upstream or downstream. You can just put it on the N terminal of the protein. You can put it on the C terminal of the protein. And most of the time, the protein is perfectly functional, even with that, because it's very short. Sometimes it may not, but most of the time. So now you have a protein by the recombinant DNA technology, and then you express that protein with this tag right here. And now what you do, that all you need is an anti-epitope antibody. That's it. You just commercially available. Suppose this is called a FLAG tag. So all you need is an anti-FLAG antibody, just one, commercially available, and that's it. So this molecular biological process, this whole thing can be done just in 1 week, and even less than that, and you have your protein tagged. So your experimental process actually really becomes faster compared to raising primary antibodies against that. But on the other hand, there are many primary antibodies are commercially available to the proteins which are widely used though. So here, classic example of this, entirely different proteins, different cells. This is nothing like a co-localization. Right here is the MYC, which is added to this font C2, and this is a FLAG. And you can see that they are all attached with a red dye. That's why you are actually seeing. So you can actually take a look at this, and you can actually look at co-localization of the different protein, which is not shown in this picture. So don't get it wrong that this is not co-localization, because remember, if you just look at the cells, these are different cells, these are different cells. They don't look separate. But you can have a picture where you have the same cells that is attached with this red dye, And in the same cell, you can have another colored protein which will show up this like a green or blue color or a yellow color or anything like that. So here the nucleus is stained blue by either DAPI or Hoechst. So this is just an idea that how the epitope staining can work. All right, first clicker question. Everybody good? All right. This is the last easy question. So it will get a little bit stiffer probably, but that's it. Okay, all right. So you need the dichroic mirror, and that's the correct answer. All right. Now the next one is a little take your time. You are interested in performing indirect immunofluorescence microscopy in a virally infected human lung tissue using an antibody raised in goats. That's your important part. That can detect the interferon. Which of the following antibody will work as the best secondary antibody in this experiment? Good. So that's correct. So rabbit anti-goat. So again, remember that whenever you see that it is raised in the animal, remember that you should have an anti before that animal name. And it should have the where you are raising should be something other than that animal. So that's the short way to do that. All right, good. But some of you actually took the goat anti-human conjugated rhodamine as well. Remember that you are you are detecting it in a human being. So when you are detecting it in a human being, then you cannot have an anti-human for that because you have raised already. I've given the term that you have raised the antibody in goat. So you are detecting the protein in the human being, and it should not be goat anti-human. All right. So now let's take a look at the next part. The variation 3 is the localization of the proteins in the different cell, and this is probably very, very popular, and a lot of people actually already know this. This is from the Cura jellyfish. You actually get this green fluorescence protein, which is a barrel-shaped protein. You're looking at the molecular structure from the side. It's actually a beta barrel-shaped structure, and you have some aromatic amino acids in the middle, which actually cause this fluorescence. And it is very close to a common fluorescent dye, which is called fluorescein. It excites at 492 and emits at 525 nanometer. So blue is in the kind of excitation range, and then green is the emission range. So now this is the beauty of this. And many of these are engineered proteins. People make fun of these because they just constantly mutate and then change. And what you are seeing that these EGFP, ECFP, these are called enhanced proteins. So your EGFP just has a mutation of one amino acid that actually enhances the fluorescence. And now changing these different amino acids or aromatic amino acids, the scientists have actually developed all these different colors, and you can actually attach these to the proteins to have, just look at that multicolored cell pictures or something. You will almost have all these, like it looks like almost like an artwork, and it really is. So here is really an artwork. So here, what you are seeing, this is a agar plate. I'm sorry. This is an agar plate. So what you have put some bacteria. Each of the bacteria is expressing one of those fluorescent protein from the previous picture. And pretty much all of these are actually excited at the ultraviolet range. So that's why it's black. So you put under a black light, and you will see that the whole of this thing will fluoresce. Now, remember that one of the important thing I also said that in the lecture video that I uploaded, You can visualize this with your naked eye with all of them fluorescing the same way. You do not have to put a filter in front of your eye because this is large, huge. But if you have all these different proteins in a single cell, you cannot visualize all of them because just due to that they are so close, they are going to mask each other and you have a blurred image. So you have to take all these different filters at a time. You can reconstruct the similar type of image if it is in the cell like this, but each time you have to get just one dye or one fluorescence, and then you can proceed with that process. So this is just like an amazing way that how these cells are. So the process is the same. So what you do, that it is the same way that we talked about the epitope tagging. You just add a GFP here, and this could be a RFP, red fluorescence protein. It could be a YFP, yellow fluorescent protein. It could be a cyan CFP. It could be any different kind of protein, whatever you look. You just put that gene upstream of the gene of your interest, and you produce the protein as a fusion protein. So usually these proteins are in between about 25 to 30 kilodalton protein. Sometimes it may compromise the function of the protein. That's very unfortunate, but many times it does not. It's just happy in there, and it just produces as a fusion protein, and its physiological function can be it's physiologically completely functional. So what you can do, that you can track this in a living cell. This is the advantage of that. So why this is important, or the variation over your antibody or based technique, remember that in the previous technique, in the indirect immunofluorescence, you have to kill the cell, fix it, permeabilize the membrane so that the antibodies, huge large molecules, they can go in and bind to the protein. Here you are using this in a live cell. So that's the advantage, that you express this protein in a live cell. And here you can see that these are some of the membrane vesicle of Rab8, and you can see that on the right hand side, this is a picture from your phase contrast. This is a picture from fluorescence. Gives a lot of thing, and you can see that right here, probably this black area, that is probably the nucleus, and you can see it kind of right here, nucleus. That the membrane vesicles are not in that area, so you can actually see that. But still, if you see that fluorescence image, it does not give you that much of information. It is fluorescing, that means Rab8 is all over the cell, but it can be improved. So on the other hand, this gives a very nice idea about something. So remember that this is an amoeba, same thing, a non-pathogenic amoeba, which does the endocytosis. Now this is from your Bio 101, that endocytosis, two types. You have a phagocytosis, cell eating large pieces. Then a pinocytosis is picking up either liquid droplet or small molecule. When they pick up these small things, they actually form, right here is a pinosome. So pinosomes are just endocytic vesicle, just smaller in size than a phagocytic vesicle. So here, the actin is actually labeled with fluorescent, with a GFP. So ABD is an actin binding protein, which is fused with the GFP so that it can bind to the actin, and the actin will light up. Now remember that these are all black and white images. So the bright area that you are seeing right here, these are all actually fluoresce green in a colored microscopy. But this is just a black and white image. So here you can see that a pinosome is forming right here, and you have a high amount of actin right here. And now you can see that the vesicle is actually invaginating. The complete pinosome vesicle actually formed, and then once it's pinched off from the plasma membrane, so everything is internalized. Now here you can see the actin is disappearing. The vesicle is still there. So the idea is that in the endocytic process, actin plays a very important role. So this is actually being shown beautifully by this process. So very powerful technique using this, and you can see this in a live cell. That's the advantage. So fun pictures. You actually can buy probably these in Walmart, PetSmart. Have you seen those with the black light, all these fluorescent? These are actually recombinant transgenic fish, actually, commercially prepared. You can have transgenic mouse. They will fluoresce under the black light like those. A jellyfish could be natural as well. But tobacco plant, you can just make anything, anything like that. One of my friends, he made some caterpillars which actually fluoresce. So you can just easily make these transgenics with that. So this is just for fun. All right, the next part, which is that that's wrong. So that's no, that's correct. That's the 2C is the fluorescence microscopic technique, and we will start with this, which is the deconvolution microscopy. We are not still going into the confocal on the direct one. This is still the fluorescence microscopy, but what you actually do, that you actually use the computational power so that you actually get the focal point, and the computer actually, again, you just get the overview of that. You do not have to go into the engineering or physics of that, but it can actually reconstruct the image from every single plane so that all the other out of focus lights are actually eliminated. And when you actually put the image together completely by computation, you actually get this three-dimensional image. So you can see that our regular fluorescence microscopy, an image like this, but just by this deconvolution of fluorescence microscopy, you can get a much clearer resolution. And these are done completely by an algorithm which is actually being developed constantly. So each photon that actually hits the camera, it can actually tell that whether that photon is coming from an out of focus area or in focus area. And these are the very complex algorithm by which you can actually improve the image as such. So here, I'm not reading the slide, so let's take a look that the advantage on top of this is a confocal microscopy. And confocal actually uses the same technique that I said, that when you have the fluorescence microscopy, you have a sample and you have a point of focus, but you are getting light or the emission light above the sharp point of focus from below the point of focus. That's the reason the images are blurred. Here what you use the excitation source as a laser, different types of laser, red, blue, black, violet, and the laser actually through the dichroic mirror, there is a scanner. So we'll come to the scanner in a second. But the basic idea is that you have a focal point, which is the blue line right here. And that light will the same way will emit through right here, and it will go through a pinhole, and that is the only light will reach the detector. So on the other hand, you can see that right here, the green line is the out of focus emission, and it will be blocked by that pinhole. It will not reach the pinhole. So this is basically a series of pinhole. You just illuminate just a small area, and that gives you a much clearer image. And we will talk about the scanner mirror in a different picture. We will come back to that what it does. So here, this is a clearer idea that how this actually works. So you can easily see right here, this is actually the 3D specimen, and this is the point of focus. Now, what happens by the pinhole, you can see that if this is the point of focus right here, the pinhole only allows this light to come to the detector or the camera, whereas if this is up or down, you can see that it is blocked by the pinhole. So that's the beauty of the system. That is very precise. It's just a point. Excitation light is coming from a very tiny point. So you can easily see now the improvement. You have a conventional fluorescence microscopy versus you have the confocal microscopy. I mean, like, you can look at the just confocal microscopic images, just give a Google search, and you will be amazed by seeing the images. But there are many improvements, but still confocal was, last 20 years, is probably the workhorse of many laboratories to clearly see images as. So here we are looking at two different types of confocal microscopy, and we will talk about that scanning mirror first. So what you are seeing right here is right here you have a laser source, and that laser through right here comes to a scanning mirror. There is an X and a Y mirror. What they do, that these scanning mirrors actually go back and forth. So again, let's first take a look at what it's actually doing, then I'll tell that. And it's falling on the image, and the light, which is the emission light, is going back right here. It's going through the pinhole to the photomultiplier tube, and you actually record the image. Now what happens that each time that X and Y mirror actually moves constantly. So what it does, that it actually makes the scanning in a raster fashion. It just goes up, turns, goes up, down, up, down. It constantly, as you can see right here, in a raster-like fashion, it will scan the whole image from one end to the other. So each time you are getting a precise focusing point from this point scanning, and you actually merge all those images for the clear picture. So that is your point scanning. It is there's a drawback. Gives you nice 2D, 3D images, but the drawback is that it is very time consuming. So on the other hand, right here, that what you are seeing, that this is a spinning disk, like the spinning disk laser scanning microscopy. Here you have a pair of pinhole disks. So the first one is a set of lenses. The second one is actually the pinhole. So what happens that one portion of the lens is actually illuminated, one portion. And this is, see, that not the whole, but quite a bit of a portion. These lights passes through simultaneously through the pinholes, falls onto the specimen, then it comes back, again goes back to the specimen and goes back to the detector. So what is the advantage of this compared to that? Here, you are scanning one point at one time. Here, if you just look into your book, you will see that you can scan simultaneously 20,000 points at the same time. So it is extremely fast to develop, to develop that image. So what is the advantage? That if you have a cellular process, this is your take home, if you have a cellular process which is extremely dynamic and extremely fast, it actually happens, goes away, millisecond events or a couple of seconds. That event, if you want to actually do a video microscopy, this will not give you that video microscopic image, but this will actually give you that. So you can actually record very dynamic, fast cellular images with this spinning disk laser microscopy. So here, although this is not the best picture, I will try to find some better ones of the spinning disk, but this is just this picture from your book. This is a fission, this is an yeast. And what you are seeing right here, this is a dynamics of this microtubule. You can see that this is being developed right here. It's being developed, and this is basically a budding yeast. And this is actually from about 140 millisecond, not millisecond, 140 seconds. And you can see that the dynamics of these microtubule assembly and how they changed over the period of time in a budding yeast. And this is a classic example that in a short period of time, this fast a pinhole, a typical point scanning, laser scanning microscopy will not be able to do this, but a pinhole can do this. All right, spinning disk can do that. So the next one that what we are talking about is the two-photon excitation. So here, this is an interesting part that what happens that when you actually excite any fluorescence microscopy or confocal, that so long what we were talking about, suppose this is your excitation photon, 488, suppose that's in the blue range. It goes right here, excites the electron, it just become loose and emit photon at 507 nanometer, and then it goes back to the ground state. And again, I mean, like not going into the chemistry, but this is just how it works. But remember that this 488 nanometer is an extremely strong visible wavelength. That is your most important part. It's an extremely strong visible wavelength by which it is actually being excited. In a two-photon microscopy, what you do, you use an extremely fast laser pulse, but if you look at the photon, the wavelength in a 960 nanometer, can you see 960 nanometer? What is your visible wavelength approximately? Say I'm like 400 to 750 or something like that. So it is at the infrared range. So you cannot actually see that because it's an infrared, but still there are fluorophores which can be excited by this infrared as well. It doesn't have to be a visible light, but there is something different with that. It has to be hit by 2 because this actually do have, these photons have half the energy compared to a 488, close to half. So it has to be hit by 2 protons, which 2 photons, one here, another here. And these 2 photons need to hit within a femtosecond range. So it need an extremely sophisticated laser pulse, which will actually put that particular 2 photons onto the fluorophore within no time, almost simultaneously 2 photons. If that is done, it will fluoresce the same way. So what you can actually completely emit from here is that the light that is being scattered from this excitation visible range light. And a classic experiment is shown in right here. You can see that this is a glass cuvette, and the liquid that is in there is actually a fluorescent material. And you can see that right here, 488 nanometer, you have such a bright cone. And when you are having the excitation, you can see that due to this, it is not very clear. I mean, like, you have so much of bouncing light around, you cannot get a clear image compared to that. Here, see that it's in an invisible wavelength because it's infrared, but you can see that the point where the fluorophore is actually completely fluorescing at the single plane. You can just appreciate that how clean this is, and this is your 2-photon excitation microscopy. So where you actually utilize this? So you can actually look at a live specimen. You actually inject a live specimen like a mouse brain with a dye, and you can actually image almost 1 millimeter. I mean, like, in microscopy, 1 millimeter is like Earth to sun distance. It's a huge distance as such. So you can be, your tissue could be 1 millimeter deep, and you can visualize by this 2-photon microscopy because the fluorescence is coming from just only one plane, and the thickness of the specimen will have no effect. On the other hand, you just cannot do that with this conventional. Think about that your tissue specimen is probably something like this. So anything is, I mean, like, you will have a completely blurred vision. You cannot just see that anything from here. On the other hand, this is so much clean. You can use your filters to cut off most of the material, but still it is not as clean as a 2-photon excitation microscopy. So this is, again, you can see that these are some of the lobster neuron in live lobsters. You can see that they're taken at different times, at different, they're stained with a different fluorophores, but you can see that the long accents of the neurons. So the last one of the modification of this is the TIRF, or the total internal reflection fluorescence microscopy. So you have seen that most of the fluorescence microscopy, or most means like all of the fluorescence microscopy, your incident ray actually comes, or the excitation beam comes through the objective, and it goes back the same way through the dichroic mirror. So it's the same thing, it's nothing different. But what happens here, that here the focusing point is so precise, it actually only illuminate an extremely small part, 50, 100 nanometer, maybe up to 200 nanometer at the most, thickness from the top of the cover slip. So you have the cover slip, and the light will only go to up to 50, 100, or 200 nanometer, and the rest is going to be completely dark. So this is called an evanescent wave, and this evanescent wave, or the plane. So the meaning of this word is basically to disappear, to vanish. So basically what happened, that after this small tiny layer, the light actually vanishes. So there will be no light from the other part of the specimen. It will only light up the material which is extremely close to the cover slip. So what you can see, that in a spatial arrangement, whether the material is close to the cover slip or distance from the cover slip, you can get that information. So let's take a look at a picture, so that will tell you that how it is. So this actually gives you an epifluorescence that we actually see. Epi means from the above, and it will just illuminate all of the specimen just like this right here. On the other hand, you can see that right here is the evanescent field, where it just only fluoresces or sends the light to the layer closest to your cover slip. All other areas are not illuminated. So you can see that if you just use your regular epifluorescence, the regular fluorescence microscopy, from the above, these are the cytoskeletal elements, and you can see that they are all light up pretty much. You cannot have any information that where in the 3 dimension they are. Are they close to the cover slip? Are they deep? You cannot tell. So when you go to the tear microscopy, you can easily see that these are the one which is closest to the cover slip. This is in the advanced field, whereas right here they are much deeper. This is pseudo color, so you can see that this is TIRIF, this is epi. So this gives you an idea that how close. So what is the advantage of that? Think about that. You are growing some of the live cells and you want to image, and you want to look at a protein which is at the cell surface on the plasma membrane, which is sticking to the cover slip. So to identify these proteins where they are or where they are spatially on the plasma membrane, this is a very powerful technique. All right, clicker question 3. You're trying to track a microtubule assembly in a cell using a GFP tagged microtubule. Process is extremely fast, dynamic cellular process. What you will use? I think we are good. All right, we are good. So let's take a look, and that's the correct answer. There's no wrong answer. That's great. That is correct answer. So the next part that we are going to talk about is the fluorescence recovery after photobleaching. So what you actually study by this, and the first, before we go into that if you actually have any kind of a fluorophore, if you shine any kind of a excitation light on that, after some time there will be no electron that it will emit or relax to emit the emission wavelength. So what will happen that it will not fluoresce anymore. So this is called the photobleaching, and this is being exploited to study several different things. So what we are trying to do here, suppose here is a plasma membrane protein, transmembrane protein. These are the green on the plasma membrane, and they are tagged with, say, a red RFP, red fluorescent protein. They're tagged right here. And what you do that at a tiny point, a specific point, you actually constantly excite them for a longer time with a certain wavelength, which is the excitation wavelength, and you actually bleach them out. That means that they will not produce any kind of red fluorescence. You can see that all of the cell is fluorescing red, but you have a white area in the red. That area is bleached. So that's basically what you observe. Now what you do that you take the excitation wavelength away, and what happens that if that protein is mobile or it's moving on the cellular surface, it will recover that bleaching. But it means that the proteins from a different area will actually crawl up and then they will move, and you can see that there are some bleached molecules, but the molecules from the other areas will come in and they will restore the fluorescence. So you can see that there's a restoration of the fluorescence. So this is the fluorescence recovery after photobleaching, can allow you to actually look at the dynamic molecule which are moving on the cell and how they are moving. So you can quantify them as well. So here, this is later in the biomembrane that when you study that, we'll talk about that. But this is just an other area that you can see that on the cell surface, this is the area which is photobleached. And then you allow it to recover, and now the restored, the molecules will be restored because molecules from the other area which is not photobleached is coming to that area to restore the fluorescence. So that's the technique. So here, on the other hand, you are seeing dynamic process which you can actually quantify two different proteins. So this is an ezrin GFP. These are kind of called an ERM family of proteins. These actually bind to actins. And the same one is the EBP50. These are, bless you, these are also same. These are all cytoskeletal binding proteins, form scaffolding from many of the channels. But anyway, the idea is that what you are seeing that here you have a small area of interest and that you want to photo bleach. And that area, this is just like a schematic, and that area, suppose this is the area. So you can see that this is your Ezrin GFP. You have the fluorescence, and you photo bleached. It is completely black, still black, coming back to some extent, not much. On the other hand, if you can see right here, this is the ground state. Then you have photo bleached it, and it is recovering that fluorescence much, much faster. So if you plot them, what you can see that your right here, your Ezrin GFP, which is the left panel, is after photo bleaching, it takes a longer time to recover, whereas your EBP50 recovers the fluorescence much, much faster. So you can actually look at that how fast two molecules are moving, or the dynamic state of two molecules or even more on the cellular surface. So a very, very powerful technique that you can actually look at the molecular dynamics by using this technique, and we call this as a FRAP technique. So the next one is the FRET, which is commonly, I mean, like most of the people, I don't know why, it's the original name is called the Forster Resonance Energy Transfer as per the discoverer, but all the time people call it is a fluorescence resonance energy transfer, which is correct as well, I guess. There is nothing wrong, but they are interchangeably used all the time. But again, FRET is Forster Resonance Energy Transfer technically. So here is a very powerful technique, which I probably use that very powerful technique after each and every slide, probably. I know that, because they're really exciting. Here, the CFP, the CFP right here is a cyan fluorescent protein, and you have an excitation wavelength and you have an emission wavelength. YFP is another protein, which is a yellow fluorescence protein, and the classic thing is that this CFP's emission wavelength is the excitation wavelength of YFP. That's the key. Again, repeating, the emission wavelength of the CFP is the excitation wavelength of the YFP or any other molecule. So this is called a donor molecule, which can excite another molecule, which is called an acceptor. So what happens that if two proteins are tagged with these kind of molecules, and if they are interacting, means that they are closer to 10 nanometer, 100 angstrom. If they are that close, then the first donor molecule can actually transfer its emission energy to the second one, and it will actually fluoresce. So what will happen that if the proteins are not interacting, you will have only blue fluorescence, or the emission of the blue range. If it interacts, it will change to the yellow one. So you can tell that if there are two protein partners, whether they're close enough or not, and whether they are actually interacting or not. So that's the beauty of the thing. So here you are looking at the specific wavelengths of the YFP and the CFP, and X and Y are the two partners of the protein. Same thing. Now, CFP is actually excited at 433 nanometer. It excites at 475 nanometer. And if X and Y actually are close together due to FRET, YFP will actually emit 530 nanometer. So what you are seeing that this is actually a migrating cell. And you can see that right here, we are measuring a specific protein interaction. And on the surface, that this is a fibroblast, which is on the area where it is actually moving. That area actually have two different proteins which are interacting with each other and showing the FRET. This area, there is no FRET. That's why it is the blue area. Here, two interacting proteins are interacting with each other and showing that yellow fluorescence. So this is actually, you can actually study many different types of protein-protein interaction by this powerful technique. So here is another way, and this is actually a classic experiment where you can actually use some kind of sensor mechanism as well. So here, there is a mechanism by which you can actually measure either the cyclic AMP concentration in a cell or protein kinase activity in a cell. So how you design this experiment, you actually do have a protein which is a ligand binding domain, which is actually, by recombinant DNA technology, you add a CFP to that cyan protein, and then you have a sensor domain which is attached to a YFP. So what happens is that the sensor domain, you actually just have this tiny bar right here. That's actually a phosphate binding domain. So if this sensor domain is phosphorylated, means a phosphate binds in here, this will interact with this. So without phosphorylation, the sensor and the ligand will not bind. So what happens that this is from your basic biochemistry, that your protein kinase, any kinase phosphorylates a protein, adds a phosphate. So you can see that if the reaction goes forward, the protein kinase will put the phosphate, and if it puts the phosphate, it will just attach to the ligand, and you will have a FRET. On the other hand, the phosphatase is another protein in the cell which removes the phosphate. Kinase adds phosphate, phosphatase takes away the phosphate. So phosphatase, if it removes the phosphate from this sensor, it will go back to this state and it will become blue. So the experiment that is being shown here, that when the cyclic AMP level in a cell actually rises, the protein kinase A activity goes up. And again, this is just a real scenario that how you actually do this. Now there is a drug called forskolin. If you add it to the cell, it actually activates or elevates the level of cyclic AMP. And what happens at that time, your protein kinase A is activated. And this is actually been shown right here. So in the 0 time, you can see that there is no activity of protein kinase. So there is the sensor domain is not phosphorylated. So you can see only the CFP emission right here. And gradually after adding the drug, as the cyclic AMP is increasing, more and more protein kinase will phosphorylate the sensor, and the sensor will bind to the ligand domain, and it will change the fluorescence towards the yellow range. So this is over the period. You can see a drug activity by a sensor molecules. These are very powerful tool by studying different types of drug activities in different cells as well. So the forskolin, this is in the chart that we talked about, that we use in the class. Clicker question. So in the following list is an excitation and emission wavelength for different fluorescence proteins. Which pair of fluorescence protein could be used to perform the FRET? I mean, like, just remember that they have to be close. They do not have to be exact. I think that tells you pretty much everything.
Can you make it bigger?
Yeah. All right, 1 minute. Tag CFP, and where is 480, 482? So that's the correct answer. And why is the correct answer? If you carefully look at that, so first take a look that where is your emission of these? 480, do you have an excitation of close to that? So if you look at that, emission of 480 is close to the excitation of this. So right here, your tag CFP and the turbo GFP is the thing. 480 and 482, as I said, that within plus minus 5 nanometer, that's perfectly still fine. So this kind of question you can expect a little bit for your classes. All right, so here, the last few minutes we will just spend with the time. This is a super resolution microscopy at the very latest technique as such. And very, very sophisticated. Don't have to go into the physics. Just have 2 or 3 lines that what it is used for, and we'll quickly go over that. So the first one that what we are looking at is called the SIM, or this is actually the structured illumination. So what happens here, the excitation source actually goes in different direction in a fashion which gives you a kind of like a Moira pattern. Moira pattern is nothing but you have seen these black and white lines, and it's used to make these interference pictures as well. And when you actually collect all these illumination patterns in different orientation and put them together, you actually get an extremely detailed image. And you can see that you can push the limit from the confocal microscopics to 200 nanometer to half, 100 nanometer. So this is your super resolution microscopy. And you are not changing the laws of optics in here, but this is done by computationally using a fluorescence optics, which is the regular optics. Here, on the other hand, the stimulated depletion microscopy, or the STED, what it does, you have an excitation laser point, and you can see that this is actually diffuse. So this is diffuse, and you have a depletion beam. Remember that this donut-shaped depletion beam, this is not a physical beam, but this is a canceling light which will actually surround that excitation point. And now you have an effective excitation which is extremely precise, and by this you can, by the STED, you can actually push the limit to about 30 nanometer. So you are actually challenging almost electron microscopy, or we call this now is a single molecule microscopy, and it is an extremely powerful technique by this process as well. The next one is the photoactivated localization microscopy. So here what happens, that at a certain time you just only pulse a quick excitation, and at that time some molecules will be excited, some will be not. Whatever is excited, the computer actually takes that image. Then you send another pulse, a couple others will be excited, then other. So you take these thousands of images, and some molecule which could be close together, it could be close together, two molecules, but they will, if they light up simultaneously and if they're very close together, you cannot actually separate them, because you can see that this is a Gaussian curve that shows the distribution of the intensity. And if there is a Gaussian curve right here, see that the base will actually merge and it will look like a single point. But if you take two images where just one will fluoresce, in another other will fluoresce, you can actually find that middle point from two different pictures, because at one time this is illuminated, you can find the middle point. One time this is illuminated, you can find the middle point. When you merge the image, you actually only take the very middle point, and then if they are close apart, you can actually differentiate these 2 points computationally. So that is your essentially photoactivated localization, and you can see that how the point scanning confocal over the palm image. And the last one that you have is a super resolution light sheet microscopy, where the light doesn't come through the objective. That's a common thing. The specimen is illuminated from the side in all different directions, and many times this is a live specimen. And how this is actually done, this is a classic example where you can see that this is a green fluorescence protein, and a sensor molecule like a calmodulin, which is a calcium binding domain, is attached to it. And this is a C-terminal domain of the green fluorescence protein, and you have a ligand right here. When calcium concentration actually increases, these are the 4 dots which actually bind to the calcium, to the calmodulin. If the calmodulin binds to the calcium, it will bind to the ligand. And when it binds to the ligand, it can actually reform or make the functional GFP, and it will actually fluoresce. So these techniques can be actually done in this process. And here you are looking at a pseudo-colored, false colored, in a living zebrafish as such. And you can actually look at these samples directly by this light sheet microscopy. So these are some of the classic example. And last clicker question, if you have time for 2 minutes. Which microscopy uses a donut-shaped beam surrounding the excitation laser focus point to make up the very small and precise? All right, let's see. That's correct. So that's your correct answer. All right, so that's pretty much the end of your, we'll talk about the electron microscopy and everything.